everyone, and welcome back to another video. So today's video is Immunology and it's the immune system, and this is for EDEXCEL A and B A Level Biology. So we've got a lot of content to cover today, so I'm not going to read through the specification points, but just the main sort of ideas of what we're going to be discussing are non-specific responses, roles of antigen and antibodies, the differences between B cells and T cells, the routes pathogens take when entering the body, and also the barriers that the body has. And then for EDEXCEL B, we're going to be discussing types of white blood cells, the humoral immune response, the cell-mediated immune response, and the role of T and B memory cells in the secondary immune response. So we need to know the major routes that pathogens may take when entering the body. So I just thought I'd start by discussing what a pathogen is. So a pathogen is any organism that causes disease. So pathogenic microorganisms include bacteria, fungi, and all viruses are also pathogens. Infectious disease is a disease caused by pathogens. So this develops in the organism and it causes many symptoms which can actually lead to death in many cases. So bacteria, for example, these are single-cell prokaryotic microorganisms and they're only a few micrometers long. And viruses, these are microbes but they're not cells. They are nucleic acids surrounded by proteins, and these are much smaller than bacteria. So for the major routes pathogens take to enter the body, these can include cuts in the skin, so any injury or break in the skin can allow pathogens to enter the body. Also through the digestive system, so if you eat or drink anything contaminated. You can also inhale pathogens through your nose or mouth via the respiratory system, and also other mucosal surfaces such as the inside of the nose, mouth or genitals can hold pathogens. So the first barrier to protect us from infection is the skin. Now the skin contains keratin, which is a hard protein. It's on the outer layer of the skin, and this is what is effective in preventing the entry of microbes. So when the skin is cut, wounds do enable the entry of pathogens, and it's the mechanism of blood clotting that seals the wound and reduces the number of microbes that enter the body. We then have skin flora. So skin flora are large numbers of harmless microbes on the skin, so they prevent colonization of other bacteria, and they occur naturally, and because they live here they are well adapted to the environment. So this means when they compete with other bacteria they are suited to the conditions which are created by salty sweat and chemicals, but other bacteria are not as suited, which means that they are unlikely to survive. We then have stomach acid. So the stomach actually produces an acid called hydrochloric acid, and this kills the majority of pathogens that enter the stomach via the mouth and then travel down the esophagus. We then have gut flora. So these are similar to skin flora in that they are natural microbes that live within the gut, and unfortunately some pathogens, they survive in the stomach and they pass on to the intestines. So it's important for these gut flora to compete with the pathogens that enter the gut in order to make sure that they do not survive. Another barrier is lysozyme, and lysozyme is an enzyme found in lysosomes. So this is released and it kills bacteria by damaging their cell walls and causing bacterial lysis, which is the bursting of bacteria. So lysozyme is found on mucosal surfaces, so in the eyes, mouth and nose, and also in tears and other secretions such as saliva and mucus. So what happens when pathogens do bypass our defences and get into the body? So initially we have a non-specific response, and this involves inflammation, production of interferons, and a process called phagocytosis. So inflammation. So as we said, when we get cut on our skin, this enables microbes to enter the body. So initially it's the blood clot which seals the wound, but it's the inflammation at the site, known as the inflammatory response, which helps to destroy any microbes that do invade and do enter the body. So the inflammatory response has a series of steps in its process. So it begins with immune cells recognising foreign antigens on the surface of the pathogen. So antigens are proteins which are unique to cells. So immune cells are able to recognise this antigen as foreign and therefore identify that this molecule is a pathogen. Then damaged white cells and mast cells, which are a type of white blood cell, which are found in connective tissue below the skin, release a chemical which is called histamine, and histamine triggers inflammation. So histamine causes two things. One is vasodilation, so this is the widening of blood vessels around the site of infection. So this therefore increases the blood flow to the site. It also increases the permeability of capillaries, so the cells in capillary walls separate and the vessels leak. So these two factors cause plasma fluid, white blood cells and antibodies to leak from the blood into the tissue, and this is what causes swelling, which is known as edema. And then finally, white blood cells come along and destroy the pathogen. So another non-specific response is interferon production. So when cells are infected with viruses, they produce proteins called interferons, and these help to prevent viruses spreading to uninfected cells. So the way that interferon prevents viral replication is by inhibiting viral protein synthesis. So it activates cells which are involved in the specific immune response, and these kill infected cells. They also activate other mechanisms of non-specific response, such as promoting inflammation to bring immune cells to the site of infection. So there are many types of white blood cell, and these include phagocytes, which are macrophages, neutrophils, and monocytes as some examples. So neutrophils and monocytes are able to leave the blood by squeezing through capillary walls. Neutrophils then ingest and destroy bacteria, and monocytes then become macrophages, which engulf bacteria, foreign matter, and cell debris. The second type of white blood cells are lymphocytes, and these are T lymphocytes and B lymphocytes, and these are known as T cells and B cells. And these are involved in the specific immune response, including antibody production and immunity. There's also other white blood cells and mast cells, which we know produce the chemical histamine involved in the inflammatory response. And then for EDEXCEL B, you also need to know about eosinophils, and these respond to parasitic infections by producing cytokines which activate other white blood cells that are also called leukocytes. So for EDEXCEL B, you need to know the structure of some of these cells. So phagocytes, we've got monocytes. So these are the largest white blood cell, and they often have a kidney-shaped nucleus. We then have neutrophils. So neutrophils have a lobed nucleus, and they have granules inside the cytoplasm as well. Lymphocytes, the nucleus fills most of the cell. And with eosinophils, they have a lobed nucleus and granules in the cytoplasm as well. So the next non-specific response is phagocytosis. So this begins with damaged cells and pathogens releasing chemicals which attract the phagocytes to the site of infection. The phagocyte then recognises the antigen on the pathogen and pushes out its cytoplasm to surround the pathogen. So neutrophils arrive first, and they engulf around 5 to 20 bacteria before they become inactive and die. And macrophages, because they're larger and longer lived, destroy as many as 100 bacteria. So they're able to ingest debris from damaged cells and foreign matter. So the pathogen and ingested material is then enclosed in a cell in a vacuole, and this creates a phagosome, which is a phagocytic vacuole. Within the phagocyte there are lysosomes, and lysosomes contain enzymes called lysozymes, and these lysozymes are hydrolytic enzymes, which means they hydrolyze pathogens. So the lysosome fuse with the phagosome, exposing the pathogen to these hydrolytic enzymes, and then they're able to hydrolyze the pathogen, and any soluble useful molecules are absorbed into the phagocyte's cytoplasm. So finally, the phagocytes present the antigen of the digested pathogen on their surface, and they are now called an antigen-presenting cell. So here you can just see a little summary diagram I've made of phagocytosis to help you with the revision. So now we're going to discuss the roles of antigens and antibodies. So each type of cell is identified by a specific molecule, and these specific molecules are proteins called antigens. So an antigen is defined as part of an organism or substance which is recognised as foreign by the immune system. So this antigen is the protein that causes the immune response. So antigens can be proteins or glycoproteins, and each protein may have a number of antigenic regions, and if it's a whole organism, it will carry many different types of antigen. So the specific immune response, so B and T lymphocytes, also known as B and T cells, are very important in the specific immune response. So here I've created a summary table to show the key characteristics of these cells. So both are created by bone marrow stem cells. However, B cells mature in the bone marrow, hence they're called B cells, and T lymphocytes mature in the thymus gland, hence they're called T cells. So B lymphocytes are generally involved in the humoral response, and T lymphocytes are generally involved in the cellular or the cell-mediated response. So B lymphocyte clones can differentiate into memory, effector, and plasma B cells, and T cell clones can differentiate into memory, killer, and helper T cells. So before T cells can perform their role in the specific immune response, they need to be activated. So as we discussed, this begins with immature T cells being produced by stem cells in the bone marrow, and then they move to the thymus via the blood, which is where they mature. Next, these mature T cells leave the thymus in the blood and move to lymph nodes and also to the spleen. So as lymph fluid passes through the lymph node and the blood through the spleen, the T cells are activated by any pathogens present. So there are three main types of T cell that you need to know. The first are T memory cells, and these are involved in enabling the rapid immune response when an individual is affected by a pathogen a second or a third or a fourth time around. T helper cells are important in stimulating B cells when they are activated by releasing substances, and when these B cells are stimulated, they divide and they are then capable of producing antibodies. T helper cells also stimulate T killer cells and enhance the activity of phagocytes, including macrophages. The third type are T killer cells, and the clue of what they do is in the name. They destroy or they kill cells with antigens on their surface, which are recognised as foreign or non-self. So they destroy the pathogen themselves and also infected body cells. So now we're going to discuss the different stages in the humoral specific immune response. So this begins with T helper cells becoming activated. So this process begins with a pathogen with antigens on its surface, and this pathogen is then engulfed by a macrophage via phagocytosis. The macrophage then presents the antigen on its surface, becoming an antigen-presenting cell. The antigen-presenting cell then binds to the T helper cell with a complementary CD4 receptor. So on the T helper cells, there are receptors which are known as CD4 receptors. So this is the key term you need to know. So T helper cells are activated and divide, and they divide into clones of active T memory cells and active T helper cells. So we then have a process of clonal selection. So this begins with a pathogen with antigens on its surface being present, and then the antigen binds to a B cell which has a complementary receptor. The B cell then becomes an antigen-presenting cell. We then have our activated T helper cell with its complementary receptor, which binds to the antigen-presenting cell, which is this B cell, and it produces cytokines that stimulate the B cell. So then this stimulation causes B cells to form a clone of B memory cells and B effector cells. And these B effector cells differentiate into plasma cells. And these plasma cells secrete antibodies which bind to specific antigens, identifying them for destruction. So B cells' roles. So once they secrete antibodies in response to antigens, these antibodies bind to antigens and label them. So then once they are labelled, the antibody binds to an antibody receptor. This is usually on a macrophage. So the macrophage engulfs the antibodies and the pathogen, and then lysosomes fuse with the vacuole, which releases digestive enzymes that destroy the pathogen. So now we have the cell-mediated specific immune response. So this involves killer T cells. So T killer cells have complementary receptors which bind to antigens which are presented on specific body cells. So then the T killer cells divide and they form active clones and T killer memory cell clones as well. So this division is stimulated by cytokines from T helper cells. So without these cytokines, there wouldn't be enough T killer cells produced to fight a viral infection. So once they are stimulated, the T killer cells are able to release enzymes, and these enzymes create pores or holes in the membrane of the infected cell, which enables the cell to become permeable. So ions and water, they flow into the infected cell and cause it to burst, and this is known as lysis. So then pathogens within the cell are released and labeled as targets for destruction by antibodies from B cells. So each B cell actually only produces one specific type of antibody, and this antibody binds to only one specific antigen. So microbes, which are whole organisms, have several different types of antigen, which means several different types of antibodies will bind and activate them. So B cells have receptors on their surface which include transmembrane versions of the antibody molecules they produce. So the structure of antibodies. So antibodies, as we know, are proteins, and they have 4 polypeptide chains. 2 of these chains are heavy and 2 of them are light. When an antigen binds to an antibody, it forms what is known as an antigen-antibody complex. Antibodies also have a constant region, which remains the same, and a variable region, which has a binding site complementary to a specific antigen. So therefore, the amino acid sequence and the shape of the binding site is going to change and differ for each type of antibody. So essentially, it changes for each specific antigen. So we can see a visual diagram of the antibody structure here. So we have antigen binding sites, we have our light chains, and we have our heavy chains. And antibodies are protein molecules of a class known as immunoglobulins. So in green we have our disulfide bonds. So these bonds hold the chains together and the chains themselves as well. We have the constant region, which remains the same in all antibodies. We then have the variable region, which changes specifically for different antigens. So there are 2 different types of B cell clones you need to know. So these are B effector cells. So B effector cells differentiate to produce plasma cells, and plasma cells release antibodies into the blood and the lymph. So B effector cells are relatively short-lived and they last only a few days. We then have B memory cells. So these are longer-lived cells. So they remain in the body for months and even years. And this is what enables an individual to respond rapidly and efficiently to the same antigen in the future. So continuing on from that, we're going to discuss the primary and secondary responses. So the primary response is the first exposure of an individual to a pathogen. So this is the first time that B cells come across a non-self antigen that is complementary to its cell surface receptors. And because it's the first time, the production of sufficient antibody-producing cells takes about 10 to 17 days. So due to this time taken to produce the antibodies in order to destroy the pathogen, this is why symptoms are suffered. The secondary response is then when we are reinfected by the same pathogen, so the second exposure. This can also be the third or the fourth or the fifth, or several later on exposures. It's just not the first exposure. So this involves B memory cells immediately differentiating to produce plasma cells, which release antibodies. So these B memory cells are able to remember this antigen and can therefore immediately recall which antibody to produce in order to kill them. So the pathogen is then destroyed before symptoms are shown. So primary and secondary immune responses are involved in natural active immunity. So we can see from the graph here that the concentration of antibodies for a primary immune response is relatively low when we have initial exposure, and this also takes a longer amount of time. Whereas when we are exposed to a pathogen for a second time around, the secondary immune response is a lot quicker and it produces a higher concentration of antibodies. So now we're going to move on to some questions. So remember that if you want to have a go at these questions before I go over them and show you the mark schemes, then that would be a really good revision technique to use. So I do suggest having a look at the question, having a go, and then listening to my explanation and having a look at the mark scheme and comparing it to your answer. So question 1, a person receives a vaccination for measles virus. The person becomes infected with the measles virus. Explain the role of T cells in the immune system of this person. So we know that there's three main types of T cells in the immune system that we need to know, and these are T memory, T helper, and T killer cells. So since we're talking about vaccination, we know that vaccinations are involved in making sure that we don't develop symptoms from this virus. So initially, the person will have T memory cells due to their vaccination, and it's these T memory cells which are able to recognize the antigens that are specific to the measles virus. So then T helper cells are going to be able to activate B cells or T killer cells, and then the T killer cells are going to be the cells that destroy the cells which are infected with the virus and also destroy the pathogens themselves. So we can see our answer here. Question 2. Which cell produces antibodies? A. Macrophage, B. Plasma cell, C. Red blood cell, or D. T cell? So we know that A. Macrophage is involved in phagocytosis, so this engulfs pathogens, so this does not produce antibodies. We know that T cells don't produce antibodies, and red blood cells are of course not involved in the immune response as it's white blood cells which are. So therefore we know that A is plasma cells because B cells are activated, they differentiate into clones which are B effector cells, and then it's these B effector cells which differentiate into plasma cells, and then the plasma cells are the cells that produce antibodies. So we can see our answer here. Question 3. Inflammation is a non-specific response to an infection. Explain how redness and swelling occur at the site of inflammation due to changes in blood vessels. So let's have a look at our keywords in this question. So we want to explain how redness and swelling occur at the site of inflammation due to changes in blood vessels. So these are keywords here. So we know that inflammation involves the release of the chemical histamine. So this is released by damaged white blood cells and mast cells. And remember, mast cells are found in the connective tissue below the skin. So histamine is released, and histamine causes vasodilation. Remember, that's the arterioles dilating, the blood vessels dilating or widening. So histamine is released, it causes vasodilation, and this is what increases blood flow to the site of infection. So this increase of blood flow is going to cause the redness. It then also causes permeability of capillaries to increase, and this increase of permeability is what allows blood plasma to leave the capillary, entering the tissues, and cause swelling, which is also known as edema. And we can see our answer here. Question 4. Influenza virus can be lethal to mice. Scientists investigated the effects of interferon on the viral infection. They infected the mice with influenza virus, then gave them interferon. Explain the results shown below. So we've got a table with interferon dose on one side and the median survival time or days on the other side. So let's again highlight the keywords of the question. So it's investigating the effects of interferon, and then they're giving them influenza and interferon, so we've got to explain the results. So first we can say what we see essentially, and what we can see is that as interferon dose increases, so it goes from 0 all the way to 8 times 10 to the power of 5, the survival time also increases. So the mouse that has a dose of 0 units of interferon only survives for 3.3 days when infected with influenza, but then the mouse that is given 8 times 10 to the power of 5 dose of interferon survives for more than 42 days, or the mean, that's the mean time anyway, so the mice, I should say. So we can see that increasing the dose of interferon therefore increases the survival time of mice. And now we've got to go and think about our knowledge of interferon, because this is an explain question. And we know that interferon inhibits viral replication. So influenza virus, so therefore viral replication. So interferon inhibits viral replication. And then we can also say that the greater the dose of interferon, the fewer virus particles produced to infect other cells. We can see our answer here. Question 5. A person is vaccinated against pathogen A that is present in a foreign country. They visit this country and get infected by pathogen A and pathogen B. Explain why this person did not become ill from pathogen A but did from pathogen B. So let's go ahead and highlight the key words. So a person is vaccinated against pathogen A. See here that it doesn't say they're vaccinated against pathogen B. So then they are infected by pathogen A and pathogen B. So then we've got to explain why they become ill from pathogen B, but they don't become ill from pathogen A. So let's put a green under here and a red under there. There we go. Okay, so this question is worth 6 marks, and as you can see from the asterisk, this means that it's level marked, so it has to have a good structure and key scientific words need to be used. So we can see that because the person is vaccinated against pathogen A, this means that they've already experienced their primary immune response. So that means that their memory cells are going to be ready if they're infected again, which they are by pathogen A, and they can immediately release the correct antibodies in order to destroy pathogen A. However, because the person isn't vaccinated against pathogen B, this means when they're infected by pathogen B, their primary immune response is going to hit in rather than their secondary immune response. So this is why the person becomes ill, and this is why they experience symptoms. So in order to gain the marks on this question, you need to go into detail about the different cells, so the different B cells and the different T cells. So I'm not going to go over the whole question or the whole answer structure here, but if you flick back to the other slides about the specific immune response, they will give you all the relevant answers. And here you can see the relevant points that the exam board suggests that you use, and these are good starters for writing your answer. Question 6, so this is for EDEXCEL B only. So the photograph shows a blood smear. Which row of the table shows the correct number of each type of white blood cell in this photograph? So we have a table below. It says number of lymphocytes, number of monocytes, and number of neutrophils. So we need to remember the structure of our white blood cells here. So if we have a look at the table, the photograph, we can see that there are 3 lymphocytes, and the reason that we can identify these as lymphocytes is because in lymphocytes the whole nucleus takes up nearly the majority of the cell. So we can see from how dark these cells are that these are lymphocytes, and the nucleus is therefore taking up the majority of the cell. We've then got 5 monocytes. So we can identify these as monocytes because these are larger cells, so we can identify that these are larger cells within this picture, and they also can have kidney-shaped nucleuses or larger nucleuses, as we can see. We then have 8 neutrophils. So neutrophils have a lobed nucleus. It can be disjointed into 3 or 5 parts, which a lot of these are. So therefore our answer is A. Question 7. Explain why blood taken from a patient had an unusually high proportion of eosinophils. So for this question again, we need to remember the role of eosinophils. So we know that these are particularly responsive in parasitic infections. So when an individual is infected by a parasite, these cells work. So eosinophils are important in producing cytokines which stimulate other white blood cells and also cause therefore inflammation. So it's necessary to say that the body or the bone marrow is producing more eosinophil cells, and therefore more are being released into the blood, and this must be because the patient has had an allergic inflammation. So we can also say because of this allergic inflammation, it's therefore because they've had a parasitic infection, so for example tapeworms. So you can see our answer here. Question 8. Explain the advantage of using humanized antibodies, antibodies made by a mouse but specific to human antigens, in cancer treatment. So humanized antibodies are not going to trigger an immune response, as they're not going to be recognized as foreign because they're specific to human antigens. So this means that Fortunately, the immune system is not going to target these humanized antibodies and destroy them. So this means that the antibodies are able to bind to cancer cells so that macrophages can destroy the cancer cells via phagocytosis. And antibodies are also target specific, so they can target these specific cancer cells, and this is therefore reducing the need for any other cancer treatments. And you can see our answer here. Question 9. Explain why mouse anti-human antibodies need to be humanized in order to treat cancer. And we have a little diagram here as well. So I'm just going to bring the answer up straight away. So if you want to have a go at this question, pause this video now. But here we can see our answer. So there's a range of points that you could say. But basically, the mouse antibodies that are anti-human are going to trigger an immune response. So the question is asking you to explain what would happen, so how the immune response would be carried out. So phagocytosis, the humoral response, cell-mediated response, different T and B cells, macrophages, plasma cells, and things like that. And you can also do it the other way and say that humanized antibodies would prevent the immune response occurring. So you can have a look at the points here, and you can go back to the specific immune response slides. If you head over to my website, I'll upload the PowerPoint there as well, and you'll be able to go through the points. And finally, question 10. If you got this far in the video, then great job. I know this video is long, but there's a lot of content to cover for immunology, specifically for EDEXCEL and OCR A Level, but I didn't want to skip anything out. I did want to explain it properly, hence this video is longer. But anyway, question 10, describe how macrophages present antigens to T helper cells. So we know that initially the macrophage engulfs the pathogen with the antigen on, so it presents the antigen on its surface, so that's our first mark, and then the second mark is that this then binds to the CD4 receptor on the T helper cell. So we can see our answer here. So thank you all so much for watching. Please give this video a big thumbs up if you have enjoyed it. Make sure to subscribe down below and go and follow my other social media. I'm specifically most active on Instagram. My stories telling you guys what I'm up to, what videos are coming up. I also ask what videos you would like to see and what you would like to see earlier on and later and things like that. So make sure to head over there and give me a follow, and I will see you in my next video. Bye guys.